Armando Hasurungan biology and medicine videos. Please make sure to subscribe, join the forum and group. For the latest videos, please visit Facebook Armando Hasurungan. Now in this video we will look at an overview of transcription and translation in cells. It won't go into a whole lot of detail, but just so we get an idea of what is happening. So we begin by going inside a cell. Here is the membrane of the nucleus, not the plasma membrane, but the nucleus, where our genetic material is stored. And outside this nuclear membrane is the cytoplasm, which is the fluid within the cell. Now within the cytoplasm, we find many things, including amino acids. And there are many types of amino acids. There are 20 in total. Now going back inside the nucleus, we can find our genetic material, which are packaged up into chromosomes, forming chromosomes. We have 46 chromosomes in total, 23 pairs, one from each parent. If we unpack this particular chromosome, the genetic material is cleverly wrapped around these proteins called histones. So pulling out some of these histones, our genetic material here are organized, wrapped around these individual histones. Our genetic material is called DNA, or long deoxyribonucleic acid. If we unwind the DNA, we can see that the DNA is double-stranded and is made up of nucleotides. There are 4 types of nucleotides which make up the DNA. So what do we do with this DNA? Well, we can replicate it. And we do so during cell division called mitosis. And I have a video on that if you want to watch it. Here. Our DNA also contains many genes. Genes are regions in the DNA that do things for our body. For example, genes make templates for proteins and enzymes. To make proteins and enzymes for our body, first, the DNA has to make RNA. RNA stands for ribonucleic acid. Our body makes RNA through the help of an enzyme called RNA polymerase. Now, RNA polymerases synthesizes an RNA using one of the DNA strands as a template. This process of synthesizing RNA from DNA is called transcription, because we are transcribing. RNA typically has a poly-A tail, which means it has many adenosine nucleotide at one end. And on the other end, there is a cap. And we'll talk about these features later on. This particular RNA that was synthesized by the RNA polymerase is a messenger RNA, or mRNA for short. It is a pre-mRNA to be specific because it has not been properly converted to a mRNA. There are actually 3 major types of RNA in humans. These are the messenger RNA, mRNA, transfer RNA, tRNA, and ribosome RNA, rRNA. And they are all synthesized by different types of RNA polymerases. You have RNA polymerase 1, 2, and 3. But we don't have to worry about that now. Anyways, let's go back to the pre-mRNA that was just made. So now we are focusing on messenger RNA. The pre-messenger RNA, like all pre-RNAs, are made up of nucleotides as well. They also contain regions called introns and exons. The pre-mRNA then undergoes a process known as splicing. Splicing is basically when the regions called introns are removed from the pre-mRNA, but the exons, they will stay. and this will form a unique type of mRNA and therefore a unique type of protein. So after splicing, after introns are removed, this will form an mRNA, still with the poly A tail at one end and a cap at the other. Now the mRNA will then leave the nucleus through the nuclear pore and into the cell cytoplasm. So we know that the mRNA has something to do with making proteins. What does it actually do? Well, the mRNA serves as a template to form proteins. mRNA is sort of like a foreign book that has to be translated to make proteins, if that made any sense. Now let's just pause there with the mRNA and look at the other RNAs, because they are important as they participate in protein synthesis. So let's go back to the nucleus, to the DNA again. A specific type of RNA polymerase can synthesize pre-tRNA, pre-transfer RNA, through the process called transcription. The pre-tRNA is then spliced, removing the introns, and then you get a type of tRNA. The tRNA will then leave the nucleus through the nuclear pore into the cytoplasm. The tRNA will actually fold up into a shape that looks sort of like this. So what do we do with these tRNAs? Or what do the tRNAs do? Well, remember that tRNA is made up of nucleotides, not amino acids. And what tRNA actually do is that every tRNA will attach to one amino acid. Remember, in the cytoplasm there are amino acids. See, in the cytoplasm there is this enzyme called amino acyl tRNA synthetase. Amino acyl tRNA synthetase essentially attaches an amino acid to a tRNA, forming a tRNA with an amino acid. So now what we have in the cytoplasm is tRNA with different types of amino acids. Then we have a mRNA, which is a template for the tRNA with the amino acid. So what is missing? Well, the only thing that is missing is an enzyme that actually connects the two, connects the tRNA with the mRNA. This enzyme is called a ribosome, and a ribosome is made from rRNA, our last type of RNA. So let's go back to the nucleus and see how we form this ribosome. But before that, please remember that all these RNA, tRNA, and mRNA, they're all RNAs. They're all made up of nucleotides, not amino acids. So we're back in the nucleus to form ribosomes. We actually have to dig deeper into the nucleus, a region called the nucleolus. Our DNA in the nucleolus have genes to make ribosomes. A special RNA polymerase called RNA polymerase 1 will synthesize rRNA, ribosome rRNA. And if I had not mentioned earlier, the RNA polymerase that synthesizes tRNA is RNA polymerase 3. Anyways, let's go back to the nucleolus. RNA polymerase 1 will synthesize pre-rRNA, pre-ribosome RNA, which will be spliced to form two rRNA strands. One is small and the other one is big. The small and the big rRNA strands will eventually form the small and large subunit of the ribosome. These strands will then leave the nucleolus, where they will fold to form a small and large subunit of the ribosome. The subunits are still termed pre-ribosome in the nucleus. The pre-ribosome can then leave the nucleus into the cytoplasm. Within the cytoplasm, the small and large subunit will attach to the cap end of the mRNA, like so. So here we have a functioning ribosome with its large and small subunit attaching to the cap region of the mRNA. The ribosome has 3 special sites within it. The E, P, A site. Now each of these means something, but just for simplicity, you can think of A as attachment, E as exit. So anyways, the ribosome will move towards the poly A end of the mRNA like so. During its movement, the ribosome will act like a factory for protein synthesis. It will attach tRNA with mRNA essentially. Remember, the tRNA is made up of nucleotides, so all these are nucleotides. 3 nucleotides down this end of the tRNA is special because it is what will bind to 3 nucleotides on the mRNA. These 3 nucleotides are called codons in mRNA and anticodons in tRNA. So for example, if this tRNA anticodon matches this codon of the mRNA, it will bind on the A site of the ribosome. The ribosome will then keep moving, and then the tRNA will then be on the P site of the ribosome. The ribosome will release the amino acid from the tRNA in the P site. And then as the ribosome keeps moving, the tRNA then will exit through the E site. The amino acid that was released from the tRNA will join with other amino acids from other tRNAs to form a polypeptide or protein. Now this whole process is known as translation. So what the ribosomes are doing is that the ribosomes are translating the mRNA to make proteins. Now just for your interest, this protein that is just synthesized by the ribosome, or any other proteins for that matter, can be broken down into amino acids again, and then the cycle can continue. What is important to understand here is that amino acids, tRNA, mRNA, and rRNA are critical in synthesizing proteins. Also, many enzymes in our body are made up of proteins. They're proteins. However, a ribosome is not made up of amino acids. It is made up of nucleotides. Quite unique. Thank you for watching. I hope you enjoyed this short overview on translation and transcription.